Okay, I think I will just start now and the rest we will just let them slowly stream in. All right, so good evening, everyone. Thank you so much for signing up for this J1 EOY H2 Bio revision. So really, thank you for taking some time out of your busy schedule. I know revisions must be quite tough for you now. All right, so I hope to make the 2 hours worth it for you. Okay, so just a quick last minute for everyone who need to scan the revision kit. If you need me to flash this QR code or the link again, you can drop a message in the chat. I'm just gonna drop the link in the chat as well. So okay, give me a moment. All right, so I've dropped the link for the revision kit in the chat. If you need to refer to it, please feel free to do so and download it. All right, can everyone see my screen? Is my audio okay? Any thumbs up? If not, I'll just continue. So let me, okay, good. I see some thumbs up. Okay, so let me start. All right. So if there's any point in time, if I'm my audio cut off, anything, my camera has some issues recently. Do not fret, just drop a message in the chat. If I'm going too fast or anything, let me know so I can slow down for everyone. All right, so welcome everyone. Today is for the OVERMUGGED September EOY revision, the end of year revision for your exams for H2 biology. So we're going to cover a few topics today, mainly cell bio, biomolecules, enzymes, DNA, and genomics. Now, why these topics is because in different schools have teach the H2 bio syllabus very differently, but generally the J1 topics, these are the few that overlap across the different JCs. All right, so before we start, a little bit about me. Hi everyone, my name is Max. Okay, you can just call me Max. All right, so I've been teaching for about 5 years already. I taught many levels from primary, secondary, and JC as well, but I primarily specialize in A-level H2 biology and H1 general paper. So very recently, I graduated from NUS, double major in life sciences and sociology. And just a fun fact about myself, I love to try new things. Recently, I learned ice skating and rollerblading. So yeah, okay. All right. So the agenda for today, this is what we're going to go through. I planned out this format to maximize your learning. So first of all, we'll start off with a content revision. We'll cover some of the key revision areas of the early topics that I mentioned earlier. All right, so I'll mention to you commonly tested concepts and questions. So as to help you really know what to focus on when you're doing your final revision for your promos. Next, we'll move on to some examples. So in these examples, I don't want you guys to be so focused on copying down everything, all the notes and whatever's on the slide. Do not fret. It's mainly, I want you to pay more attention to listening to me as I explain the answering process, the answering techniques, how you should go about approaching these kind of questions. All right. Next, I'll finish up with some study tips. So this is applying not just for your EOY, but also going into J2 next year, how you should approach studying. And I think these tips apply to across all your other subjects as well. So not just for bio. And then finally, we'll end off with a Kahoot quiz. So for those who stick to the end, there's actually a prize for the first place for the Kahoot quiz. All right, so it's actually a set of the curated notes. Okay, so I hope you guys stick to the end, challenge yourself, and hopefully you can, the first place, only one lah, okay, you win the curated notes for H2 bio. All right, so let's just jump straight into the content for today. If you have any questions, please post them in the chat. I'll see if I can answer them at the end of the session when there's time for Q&A. And yeah, same thing, if I'm going too fast, anything, let me know. All right. So starting off with cell biology, I want to start off with a very important revision tip for bio itself, which is that you can save a lot of revision time by studying things in comparison with one another. So what do I mean by that? I think cell biology is the perfect example. You can see on my screen, I've mentioned prokaryotic and eukaryotic cells. So beyond that, what other things you can study in relative to each other comparison? We talk about animal cells and plant cells. We talk about, for those who have learned, respiration and photosynthesis. They are very similar processes with slight differences between them. Study them in relative to each other. Mitosis and meiosis for cell division as well. All right. So to prepare yourself, this allows you to prepare yourself for comparison questions in exams because many of these question types often ask you, they don't just ask you, explain a prokaryotic cell. Most often nowadays, questions will ask you for comparison. So we talk about similarities and differences. So let's start with prokaryotic and eukaryotic. I hope this should be familiar to you. I shouldn't be teaching. This shouldn't be brand new to you. It should be more of a revision. So first of all, we talk about prokaryotes and eukaryotes, where prokaryotes have no true nucleus, eukaryotes have a true nucleus. Now, what does this true nucleus mean? It just means that if you notice for prokaryotes, the genetic DNA, okay, it is not enclosed by a membrane, which is your nuclear envelope in the case of a eukaryotic cell. So that's just what it means by true nucleus, okay? Having the genetic material, the DNA, being enclosed in a membrane, the nuclear envelope. So some of the key differences to take note will be in prokaryotes, you have your circular DNA helped by DNA binding proteins, whereas for eukaryotes, we have your linear DNA with your histone proteins, all right? In prokaryotes as well, no membrane-bound organelles. Eukaryotes have your membrane-bound. And lastly, your ribosomes. Okay, take note of these differences. So important to know, okay, a few factors to differentiate between prokaryotic and eukaryotic cells. So another one, okay, eukaryotic cells divide by mitosis and meiosis, whereas prokaryotes, we use this term binary fission. They're not the same. All right, so moving on to plant cells and animal cells. So you can notice that the bulk of your cell biology is really just prokaryote, eukaryote, animal cells, plant cells. Okay, we go into the functions, the structure, as well as the functions of the specific organelles. All right, so commonly tested cell biology questions always involve being able to interpret your electron micrographs of your cells. So be it they show you a picture of a prokaryotic cell, eukaryotic cell, plant cell, animal cell. Okay, as you can see in the electron micrograph in my slide here. Okay, tricky questions will actually give you different types of animal cells rather than the traditional one that you see on my slide over here. All right, so one tip that I have for you to practice this would be to actually Google online different animal cells electron micrographs. All right, expose yourself to the different kinds of animal cells or plant cells even. All right, be familiar with that. As well as when you encounter such questions, read the question context very carefully, because most of the time they will tell you itself in the, oh, sorry. Okay. Sorry, my video shut off. Okay, ignore that. So most of the times these questions itself, in the question context, they will tell you whether it's a plant cell or animal cell that you are looking at. All right. But in general, the same characteristics often apply across your animal cells and plant cells. All right. So a few of the exclusive organelles to take note for animal cells would be your lysosomes, your centrioles, and your flagella. All right. I just want to make a small point here for your own information. Flagella is not present in all animal cells, but present in some. Okay. Moving on, we have your plant cells. So plant cells, I want to make a note firstly. Okay, so here are the exclusive organelles to plant cells. Now I want to make a note that plant cells have both chloroplast and mitochondria, whereas animal only has your mitochondria. So don't be confused by this. I've seen students writing that plant cells only have chloroplast. All right. So I also want you to take note the difference in size, okay, between your chloroplast and your mitochondria. Most often your chloroplast is typically much larger than your mitochondria. Next, notice that I've bolded and underlined. For plant cells, we have a large central vacuole. Animal cells have their vacuole also, but in plant cells, it's more of a key identifying factor because as you can see from this electron micrograph, the vacuole actually is large, central, takes up quite significant amount of cytoplasmic volume. All right. So this is if you're asked, all right, okay, the function of the vacuole, some students tend to forget, would be to deposit waste products, store food reserves such as inorganic ions, sugars, minerals, and specifically in plant cells to retain your cell turgidity. Okay, so next commonly overlooked area in cell biology would be the relative sizes of organelles. So I know this table here should be present in some of your school's lecture notes, but most often many students tend to overlook this, but this is actually commonly tested for your MCQ. All right. So while they seldom will test on identifying or calculating the exact size or measurement, so meaning to say like, oh, you must know nucleus is 10 to 20 micrometers, right? Very seldom they'll do that, okay? But more often what structured questions will actually test, right, okay, will be that they will give you some help. So they may show you electron micrograph, but they will often give you a size, okay? So for example, they'll give you the measurement estimate, and then you yourself have to do some calculations to estimate the rough size of certain organelles or of the entire cell itself. Okay, so I want you to take note, sizes of animal and plant cells vary. So as I mentioned, chloroplasts, just now in the previous slide, I mentioned chloroplasts generally are larger than your mitochondria. So the main takeaway of this slide would be to know your organelle sizes well and do not get tricked. Okay, I've seen students get tricked by the nucleus and nucleolus. All right, I'll show you a sample question later during the practice example, so if you know what I mean. Okay, moving on. So for the cell membrane, understand what you need to focus on will be the cellular components that make up your cell membrane. So that will be, for example, your phospholipid bilayer, all right, your phospholipids, your embedded proteins, cholesterol. Okay, so for each of these components, what are their specific functions? Now, the next thing you need to take note as well is also why they refer to the cell membrane using the fluid mosaic model. All right, so I'm going to pause here and shift over to notes. All right, so as I mentioned over here, okay, so you can see here, the fluid portion of the fluid mosaic model refers to the fact that phospholipids and proteins are able to move freely within the phospholipid bilayer. Okay, so phospholipids generally and the protein itself, they often move laterally, which means side to side. All right, whereas the term mosaic, okay, over here, refers to how protein molecules are embedded and scattered across among the phospholipids. So these protein molecules, for examples, will give you your channel proteins, protein receptors, glycoproteins. So you have to be familiar with these proteins and their functions. Okay, so back to the slides. Okay, so basically, know very well the functions of the components and their overall functions of the cell membrane. Okay, so just to wrap up cell biology, okay, the key important concepts to revise, firstly, your comparison of your prokaryotes and eukaryotes, your comparison of your plant and animal cells. Now, the next part, I'm going to spend some time on it. Okay, organelle functions and relative sizes. So I've mentioned about relative size. What do I mean by organelle functions? It would mean for each specific organelle, your nucleus, your nucleolus, your ribosomes, your mitochondria, your chloroplast, what are their functions? All right, so I'm going to take some time out to firstly explain the functions of your nucleus. Okay, so back to my excerpt. Okay. So over here, you can refer to this. Okay, so your nucleus, I hope this should be familiar to you. The nucleus, in essence, contains the hereditary material or the genetic content of the cell. So in the form of your DNA, which we've already covered. Okay, so students tend to forget the subsequent functions, which is mainly that the nucleus is the site of many of your key cellular activities, such as firstly, your DNA replication that occurs during in your interface that follows by mitosis. All right. Now, the next thing that I want to emphasize on will be your nucleolus. Okay, take note, as you can see over here, the nucleus is the site of your rRNA synthesis. So take note of how your ribosomes are actually assembled. All right. Your DNA, your nucleus contains your rRNA genes, which are transcribed to give you your rRNA. This combined with your ribosomal proteins will form your large ribosomal subunit and your small ribosomal subunit, upon which they will then exit the nucleus and be assembled into your ribosomes in your cytoplasm. So take note, they are not formed as ribosomes in your nucleus itself, but rather they are assembled in the cytoplasm. But the nucleus itself is the site of your rRNA synthesis over here, as well as the assembly of your large and small ribosomal subunits. Okay? So moving on, next we want to cover your endomembrane system. So your endomembrane system, by now, what should come to your mind automatically would be the concepts of protein synthesis, chemical modification of your proteins. So these are the organelles involved in your endomembrane system. So I'm going to switch back to my notes here. All right, be very familiar with this entire process. So what you've seen here, if I were to just explain simply to you, is that they are tracking how amino acids, their movement within the cell when it comes to the process of protein synthesis. Okay, so we talk about your ribosomes, your rough endoplasmic reticulum, your Golgi apparatus, and then subsequently your secretory vesicles. So you have to take note that individual organelles, your ribosomes, RER, SER as well, your Golgi, what are their specific functions, and the movement of the folded polypeptide, okay, that is synthesized in your ribosomes and your RER. Okay, so take note of these processes. And then lastly, as I've mentioned earlier, your cell membrane structure, general structure, functions. And lastly, one more overlooked part that students tend to forget, okay, will be your transport. So know the differences between your simple diffusion, facilitated diffusion, osmosis, so forth. Okay, what form of transport, what mode of transport requires ATP and which one doesn't. Okay, so you know, by now you should be very familiar with the definition. Are the molecules moving against or down the concentration gradient? Okay, and whether ATP is required. Okay, so I hope that covers it for cell biology. Any questions, just feel free to enter it into the chat. So we move on to biomolecules. All right, biomolecules, take note, we have three main key portions to revise: your carbohydrates, your lipids, and proteins. Let's start with your carbohydrates. Okay, so for carbohydrates, you have to know very well the structure of the monomer glucose, meaning to say you need to be able to identify your alpha glucose and your beta glucose. Okay, so you need to roughly know how to draw it out. Okay, so I've seldom seen drawing questions, but the main key point is you need to know the structure. How to identify alpha glucose and beta glucose will be based off the hydroxyl group on the carbon one, as you can see over here. Now, another way, personal memory tip, one way you can remember is that for beta glucose, okay, beta, start with B, reminds you of bottom. So the hydrogen atom is at the bottom of carbon one. Okay, so if you want to remember relative to the hydrogen atom, okay, you can remember it this way. But if not, by definition, okay, beta glucose, the hydroxyl group on carbon one is upwards. Okay, whereas for alpha glucose, the hydroxyl group you can see is downwards. Okay. Next, be familiar with the formation. Be familiar with the formation of your glycosidic bonds. So you have to be familiar with your condensation and your hydrolysis reactions. Okay, just to recap, condensation is the formation of a glycosidic bond with the removal of one water molecule, H2O. And vice versa, the breaking of this bond, this glycosidic bond, occurs via the process of hydrolysis. All right, and it requires the addition of a water molecule. Okay, so take note, the involvement of water in the formation and breaking of these glycosidic bonds. Okay, next, you also need to know the different types of your glycosidic bonds. So we talk about your alpha-1,4, alpha-1,6 glycosidic bond, your beta-1,4 glycosidic bonds. How are you supposed to identify them? And what polysaccharides these bonds are present in? So when we talk about polysaccharides in your carbohydrates, you need to know your three examples shown over here in my slide. You need to know them very well by heart. Okay, so we have your starch and glycogen for your storage polysaccharides, okay, both of which are mainly made up of alpha glucose. And then we followed by your cellulose for your structural polysaccharide. So when you're studying this section, right, okay, I must emphasize that you must be able to explain how the structure of the polysaccharide relates to its function. So what do I mean by this? For example, we talk about glycogen, okay? Glycogen, you should know, is made up of alpha glucose monomers linked by alpha-1,4 and alpha-1,6 glycosidic bonds, where your alpha-1,6 glycosidic bonds are at the branch points. Okay, so how does this affect the structure? This results in extensive branching. Okay, and the overall glycogen is coiled into a very helical and compact structure stabilized by hydrogen bonds. Okay, I hope you can remember that. So the compact structure of glycogen will allow many glucose molecules to be stored in a small volume in the cell and allows for glycogen to be accumulated to form glycogen granules in the liver and your muscle cells. All right, so I hope you were able to follow closely with my explanation. So commonly tested questions for this on carbohydrates will be, firstly, as I mentioned, being able to identify your monomers, your alpha glucose and your beta glucose. So seldom they will test on drawing, but just make sure that you're prepared, okay, to be able to draw. Don't forget the 6 carbons. Okay, I've seen students tend to draw it in just one ring structure, but if you notice over here, this is an oxygen atom, so do not get tricked. Okay, next, you need to be able to explain the polysaccharide structures, your starch, glycogen, and cellulose. Okay, explain their properties as well as their functions. Okay, so I'm going to switch over back to my notes. So for those of you who need a further explanation, okay, so I hope you remember your starch comprises of your amylose and amylopectin. Okay, both are made up of your alpha glucose. However, for your amylose, they are mainly linked by your alpha-1,4 glycosidic bonds as shown on my slide here. All right, whereas your amylopectin is made up of your alpha-1,4 glycosidic bonds as well as your alpha-1,6 glycosidic bonds at your branch points. So for your glycogen, next thing, there's no amylose, no amylopectin. It's just made up of alpha glucose residues. linked primarily by your alpha-1,4 glycosidic bonds and your alpha-1,6 glycosidic bonds. But one special feature of your glycogen is that it has more extensive branching as compared to amylopectin, which results in glycogen being more compact. All right. And you can see how both starch and glycogen, we talk about terms like compact when it comes to storage polysaccharides. All right, because we want to save space, right? And we want to be able to store as many of these polysaccharides for subsequent cellular respiration, because these are sources of your energy. Okay, and then for your cellulose, we talk about beta glucose, linked by your beta-1,4 glycosidic bonds. Now, one key identifying feature of your cellulose would be, if you notice over here, the bottom one, that each beta glucose residue, each monomer, is actually flipped or rotated 180 degrees relevant to the adjacent beta glucose molecule. So this is one defining feature. Please take note and remember of this. Okay, they may test this often in your application questions. So you must be able to identify. Okay, all right. So I don't see any questions yet. I'm going to move on. Okay, so we move on to lipids. Okay, so take note, for lipids, some examples that you've learned would be, for example, your triglyceride, your phospholipids, and your sterols. So for my revision today, I want to focus a bit more on your triglyceride and phospholipids. So first of all, know the structure well. Triglycerides, okay, take note that it's one glycerol backbone followed by your 3 fatty acid chains. So what reaction is this? This should be very familiar to you. Formation of your triglycerides should be via your condensation reaction, releasing 3 water molecules to form what is this bond? Ester bond. All right. So another property for your triglycerides, as shown here, we talk about your saturated versus your unsaturated fatty acids. Okay, so for saturated, I hope you know that there are more CC single bond, and hence, okay, the more CC single bonds present, the more saturated fatty acids are present in your triglyceride, the higher the melting point. Okay, and in contrast, for your unsaturated fatty acids, okay, most often the presence is your CC double bond. So this is unsaturated, this is your saturated. So I hope you remember this and understand the properties in relation to the melting points of these triglycerides. Okay. So moving on, we also talk about your phospholipids. Okay, so for phospholipids, take note, this is more of a recap from your cellular membrane, okay, because as we learn in the cell membrane, we talk about your phospholipid bilayer, correct? Okay, so take note, one key difference here will be that instead of having 3 ester bonds formed, here, if you notice the phosphate head, what we call this is your phosphoester bond. Okay, so this is more just a revision of your cell membrane functions. Okay, so in general, you need to know as well your triglyceride function. Okay, so for example, energy storage, heat insulation, buoyancy, and protection. So these are for your functions of triglyceride. Now, I have one commonly asked question by students. If lipids have higher, okay, let me go back to triglycerides. So if lipids, okay, so triglycerides, for example, if they have higher calorific value than other respiratory substrates, such as your carbohydrates, okay, because one of the key functions we learn about carbohydrates is that they are a main respiratory substrate, right? So why is it that lipids, if they have higher calorific value, why are they not the main respiratory substrate? Okay, so my explanation to this answer would be because if you notice, as comparing your lipids, your triglycerides, as compared to your carbohydrates, right? If you notice just now when I show you your glycogen and your starch, they are pretty straightforward structures. But if you look at your lipids, right, this is more or less just a general formula. Okay, but you see lipids can get quite complicated, especially in the fatty acid chain. So if you notice this R groups here, okay, they can get pretty long and they vary across the different triglycerides. All right, so because of this, okay, triglycerides tend to be more complex structures, and our body would therefore require more chemical reactions to convert them to simpler structures to be used as substrates for your cellular respiration. Whereas compared to your carbohydrates, okay, we talk about glucose. So we have many glucose residues, right? And then they form, for example, your starch. Okay, these are much more simpler structures. Okay, they can be broken up much more easily to access glucose to act where your glucose will thereby act as your respiratory substrate for your energy. Okay, to generate energy. Okay, so I hope that is a good explanation. Okay, let me move on to proteins. Okay, so I see there's a question in the chat. I'll look at it after the session. Okay, so I'll explain later during the Q&A. Okay, so proteins, right? Firstly, as you can tell the story by now, for carbs, lipids, proteins, you need to know the monomer by heart very well. Okay, so shown here is the general formula of your amino acids. Take note of your amino group, carboxyl group, the one hydrogen atom here, as well as the the side chain. Okay, otherwise known as your R group. So be very familiar. For amino acids, what are the bonds holding them together would be your peptide bonds. Okay, so while you don't have to memorize I know there's 20 different types of amino acids, you do not need to memorize the entire structure because there's too many. But what you must understand from that is to be able to understand that different amino acids, because they have different R groups or side chains, the amino acids themselves therefore have different properties based on the R group or the side chain. Okay, so for example, you need to know whether a certain amino acid is polar, non-polar, acidic, or basic. Okay, so the bulk of proteins, right, this chapter, what I would say that most schools test on would be your protein level and structure, your organization and structure. So I'm going to take some time to explain this part. Follow closely. Okay, you need to be very clear on knowing how to explain each protein level and structure. Okay, so you have to know the structure definition, how to define it, as well as where are these bonds occurring. Okay, so let's go one by one. Primary structure. Okay, you can see here number and sequence. So what is your primary structure? It's the definition. So your specific number and sequence of amino acids that comprise a polypeptide chain. Okay, and then what are the bonds that is holding your primary structure together? It is your peptide bonds. Okay, so to put it simply, again, I repeat, the primary structure of a protein refers to the specific number and sequence of amino acids that comprise the protein, held together by peptide bonds between the amino acids. Okay, so notice that I really go step by step to explain out the primary structure, and you should be doing this for your examinations if they were to ask about any of the structures. Okay, so move on. Your secondary structure, how does it arise? Okay, your secondary structure arises from the continued folding or coiling of the polypeptide chain. Okay, so secondary structures we learn two, your alpha helices and your beta pleated sheets. Now, what are the bonds? The bonds in your secondary structure are your hydrogen bonds that are occurring between the peptide bonds. Okay, so I'm going to pause here. Let me switch over to my curated notes. Okay, so I feel that certain students tend to get a bit confused over this part. Okay, so you can see here. Okay, I hope everyone can see my screen. The secondary structure is the repeated coiling and folding of a polypeptide chain maintained by hydrogen bonds formed between peptide bonds. So notice the hydrogen bonds are between your peptide bonds. They are not between your amino acids. Okay? Hydrogen bonds are formed between the NH group in the peptide bond of an amino acid and the CO group in a peptide bond of another amino acid. Okay? So as you can see here, what I want to focus on, this is what I'm talking about. Here, I hope you can see my annotation. If you notice the dots here, these are your hydrogen bonds. They are not occurring between amino acids, but rather the peptide bond. So you can see, okay, I'm going to draw in purple, circle here. I know it's a bit messy, so try to bear with me. The purple part is your peptide bond. Okay? And notice that the hydrogen bond is between peptide bonds. So I know it can get a bit confusing. If you get really overwhelmed, try to take it slow, reread the sentence a few times, refer to diagrams such as this. Okay, it should help you better with understanding the content. Okay? Okay, so back after your secondary structure, so take note there's alpha helices and beta pleated sheets. You have to know in general how to identify them. All right. Next, we move on to your tertiary structure. Now, what is the definition of tertiary structure? Okay, it refers to the unique three-dimensional conformation of a polypeptide chain, resulting from the continued folding and coiling of the polypeptide chain. So next, remember I say, for each structure, you need to mention the definition of the structure followed by the bonds. So what are the bonds in your tertiary structure? They are held together, the unique three-dimensional conformation of the polypeptide chain, held together by your 4 bonds. Okay, let me type it out so it's better for you. We have firstly your hydrophobic interactions, followed by your hydrogen bonds, followed by your ionic bonds. And lastly, in some proteins, we have your disulfide bonds. Okay, so you can see I've typed out over here. Take note, these are the 4 bonds occurring in your tertiary structure. And where do these bonds form? They form between your R groups of each individual amino acid that comprises the protein. Okay, so over here, circled in purple. Okay, because you can see over here in this diagram, the polypeptide chain, they coil and they fold all over like that. This allows for each amino acid, the R group, to be exposed to maybe in close proximity to another, and then they can form one of the 4 bonds over here. Okay, I hope that's clear. So moving on, lastly, we have your quaternary structure. Now, as you can see here, tertiary structure, we often talk about primary, secondary, tertiary structure, we only refer to one polypeptide chain. Whereas for your quaternary structure, it refers to more than one polypeptide chain. So you can see consisting of more than one amino acid chain or polypeptide chain. So 2 or more polypeptide chains, they fold together. But what are the bonds? The bonds are exactly the same as your tertiary structure. So it will be your hydrophobic interactions, your hydrogen bonds, ionic bonds, disulfide bonds occurring where? Between the R groups of amino acids of one polypeptide chain, okay, circled here in purple, and the R groups of amino acids in another polypeptide chain. Okay, so you can see the purple and the red. All right, so I hope this wasn't too confusing. I hope you can follow me. Okay, so I know it's a bit heavy in content. So what I recommend to you is that really go step by step and understand each of the levels very clearly. Because once you really master this, okay, I think it's one of the easier sections to score. Okay, so try to go slow. Don't overwhelm yourself with the content. All right. So next, you need to take note of the some examples of your proteins. The 2 examples in your syllabus will be your hemoglobin and collagen. So for these 2, right, okay, take note of how to explain the structure. So for example, you should know hemoglobin consists of 4 polypeptide chains, 2 alpha chains and 2 beta chains. Okay, so know the structure and in relation to its function. And same for your collagen. Okay, so one more thing that I want to highlight here, okay, so please take note, application questions. So application questions, right, they often test your understanding on amino acid properties. So for example, let's say we have a protein, right, that does not denature at high temperatures. So what does that clue suggest about the protein structure? Okay, I want you guys to just keep this question at the back of your head. We'll look at a concrete example of such a question later. Okay, then if we move on to more simpler structured questions, right? Okay, structured questions will often test, for example, your identification of protein structure. So for example, they'll show you a figure, a diagram, and they'll ask you to explain which of the protein structure level it is at. So basically your primary, secondary, tertiary, or quaternary. And then you need to explain the bonds that make up the structure and define the structure itself. All right. Okay, so last part of your proteins would mainly be on your denaturation. Okay, so this actually ties into your enzymes, which is the next chapter that I'm going to cover. So take note, for example, for subsequent protein structures, your tertiary, quaternary, right? Okay, we talk about what are some ways in which we can cause a loss in your protein structure. Okay, so the loss of the unique 3-dimensional conformation of your protein will be your denaturation. Okay, so what causes this? So for example, we have your high temperatures. Agree? Okay, we also have your sometimes addition of heavy metals. Okay, so changes in pH, another as well. So know for each medium. So let's say we have your temperature, right? Okay, which of the 4 bonds are disrupted? Is it your hydrophobic interactions, your hydrogen bonds, your ionic bonds, or your disulfide bonds? Okay, which of the bonds that are affected by the medium? Okay, temperature, pH change, and so forth. All right. Okay, so we wrap up biomolecules. Some of the key concepts that I hope you guys will spend more time on to focus. Firstly, understand what are these terms, your monomers, oligomers, and polymers. Okay, so some students are confused over the term oligomers. Okay, so if you take econs, so you should know monopoly and oligopoly. Mono just means one, oligo means a few. Okay, and poly means a lot. Okay, so you can remember monomer, if you talk about monomer of carbohydrate, so what is the monomer of, for example, starch and glycogen? It will be your alpha glucose. Agree? Okay, then when we say oligomer, oligosaccharide, for example, that just refers to a short carbohydrate chain. So short, a few glucose molecules. Okay, and then we talk about polymer, polysaccharide, which means many glucose molecules make up a complex carbohydrate structure. So basically just be familiar with all these terms. Okay, so they can be like mononucleotide, oligonucleotide, monosaccharide, polysaccharide, oligosaccharide. Okay, so let's go through each of the 3 biomolecules. For carbohydrates, what do you focus? As I already mentioned, this is just a short summary. Take note of your alpha and beta glucose, how to identify the structures, okay, how to draw them, 6 carbon, all that. Glycosidic bonds, okay, what are ones that you learn? Alpha 1,4, alpha 1,6, and beta 1,4. And these glycosidic bonds are present in what polysaccharides? So we learn your structure, for your storage, we have your starch and your glycogen, as well as for structural polysaccharide, we have your cellulose. Okay, so know for each polysaccharide the structure. How does it relate to its function? Okay, next we have your lipids. So we spend more time on triglycerides and phospholipids, but also take note, I think one lipid that students tend to forget will be your sterols. Okay, for triglyceride and phospholipids, there's the presence of your ester bonds, okay, between your glycerol and the fatty acid chain. However, for your phospholipids in particular, we have your phosphoester bond. Okay, and then know in general, your triglyceride, what are the functions? Your phospholipid, what are the functions? So as I mentioned, triglyceride earlier with your higher, they are alternative source of energy, okay, buoyancy, protection, heat insulation. Whereas for your phospholipid, they are mainly the functions of your cell membrane, as we covered in cell bio. Okay. Lastly, for your proteins, know the amino acids. All right, so how to identify an amino acid, the protein level and structure. Okay, so the definition, as I mentioned, how to define primary, secondary, tertiary structure, how to answer those kind of questions, you must know the bonds that occur. All right, so I actually want to highlight one more thing about amino acids. A commonly tested question for proteins, right, will be, they'll often show you a polypeptide chain. You must be familiar with peptide bonds. How to identify a peptide bond. So where is the peptide bond in the polypeptide chain, for example? Okay, so to be familiar with this, okay, what I recommend, let me scroll back. Okay, so as I said, be very familiar with this amino acid structure, but take note, right? Okay, sometimes the R group here, right, they have a lot of amino groups or your carboxyl groups. Okay, so don't get confused. To identify an amino acid in a very long polypeptide chain, right? Okay, you always try to find the central carbon, the alpha carbon. Okay, because for each carbon, notice that this is one of the key traits. It's connected to a lone hydrogen atom. Okay, and then the side chain. So trace it out slowly. Okay, make sure that the carbon that you're looking at has one hydrogen atom. Okay, the R group can be anything. It can consist of this. But the peptide bond, okay, it must be between one carboxyl group and one amino group of the adjacent amino acid. Okay, so just go slow. Okay, and don't get tricked by the side chain, because some amino side chains have your carboxyl group amino groups. Okay. All right. So let's move on. So that is mainly it for your biomolecules. Okay. Okay, I see a question here. Hello, can I clarify? Phosphoester bonds and ester linkages are not to be used interchangeably, right? Yes, okay, good question. Yes, very important. Let me scroll back first. Okay, so you can see here, right? Ester bonds are occurring between the glycerol molecule over here and your fatty acid chain. Okay, but your phosphoester is between the glycerol backbone with a phosphate group. So that's why they specially differentiate the name. Phosphoester bond, phospho, and then the ester bond. Okay, whereas this one, between your glycerol and the fatty acid chain, this is your ester bond. Okay, so how to take note? It's from its name. Phosphoester bond comes from the phosphate group. Okay, so yes, you cannot use them interchangeably. They are talking about different things. Okay, right. I hope that answers your question. Okay, let's move on. Okay, next, enzymes. Okay, so to start off, what to focus on enzymes would be very basic. Very basic important thing is that enzymes are your biological catalysts. So most of them, right? Okay, I hope you know by now. Most of them are actually your globular proteins. But there are special cases. There are RNA molecules. So you should know RNA, ribonucleotide. there are ribonucleotide molecules that serve as enzymes. So they can catalyze reactions. Okay. Those are special, those are called ribozymes. Okay. So be familiar with your lock and key and your induced fit models. Okay. It's not shown on the slide here, but I hope they're familiar to you. But more importantly, what you should focus on would be to know the main characteristics of enzymes and their mode of action. So what do I mean by characteristics? So basically shown here, enzymes are highly specific to their substrate. Enzymes remain chemically unchanged at the end, before and after the chemical reaction. And enzymes are effective in small amounts. All right. So as I mentioned earlier, right, enzymes, because being globular proteins, okay, you can see over here, as shown in this diagram, they have they can be tertiary structure or quaternary structure of the protein. Okay, so you can see all these little balls here, right? They are just your amino acids, but they have been folded into a very unique three-dimensional conformation that has an active site. So as you can remember, what is this conformation? What is this tertiary structure held together by? Your 4 bonds. Hydrophobic interactions, hydrogen bonds, ionic bonds, and your disulfide bonds. Okay, so it's the same story all over again. Okay, so some questions, some students might ask me, okay, are some enzymes quaternary structure? Yes, that's possible. There are some enzymes that require multiple polypeptide chains to be folded together, and then in the quaternary structure, the active site is formed. Okay, so it really depends on the protein itself. Okay, so as you can see on the diagram on the right here, the next important part will be to take note, be very familiar with this graph. A commonly tested question is they will give you the regular reaction. Okay, you can see the progress of the reaction and the energy level, and then they'll ask you to draw. Okay, what does the energy level look like when it's what does the reaction look like when it's catalyzed by an enzyme? Okay, so I want to highlight over here. Where's my annotation? Okay, so be very aware. Notice that the initial state remains the same regardless of having a catalyst or no catalyst. The final state as well. So these two sections must make sure that they are aligned, that they are same and remain unchanged. What changes is that you have to clearly show that there is a lowering of the peak. Okay, so exactly this graph, be very familiar. So what is this what is happening here is basically the enzyme is providing an alternative pathway with a lower activation energy. Okay, so also don't be very funny. I know I see some students draw like that before, like a very sharp pointed peak. Okay, try not to do that. It's not, most of the time it's not this sharp change. Okay, it's more of a gradual, you can see like a soft peak. Okay, all right, so moving on. Okay, how do these enzymes actually provide this alternative pathway? So take note, I'm going to explain a few. So how does this lower activation energy come about? So firstly, we have your formation of your transition state. So how? First, enzymes allow for close proximity, okay, of the substrate. Next, they also ensure your correct orientation of your substrate, your reactant molecules, all right? Then sometimes enzymes, what they do is they will destabilize the bonds that are holding the reactants together. So let's say we have an enzyme that is catalyzing a reaction that breaks up a big molecule into a smaller one. What the enzyme will do is that it will destabilize the bonds that are holding the reactants, holding the substrate together. Okay, how? Because you don't need to know the exact process, but some enzymes, what they can do is they will distort the structure of the reactant or the substrate. Okay, this will affect the binding. Okay, and then hence causing what we will call strain. All right, and then because this strain, there's this strain on the bonds holding the reactant molecule together. Okay, then it's much more easily broken to give you your products. Okay, and lastly, one more way is that the enzymes, they can provide a very suitable microenvironment for the reaction to occur. Okay, so these are just some examples of how enzymes will provide this lower alternative pathway with lower activation energy for your reactions to occur. All right. Okay. Next, very important. Okay, please remember this part. Okay. Next part is your factors affecting your enzyme activity. So shown here, I've just condensed into 4. Your substrate concentration, enzyme concentration, temperature, and pH. Okay, I just condensed into one slide the 4 different graphs. What to be familiar with? Be familiar with each factor, the explanation behind it, and how to interpret each of these graphs. Okay, so these graphs should not be foreign to you. They should be in your lecture notes. commonly tested structured questions, okay, they love to test on this part. Okay, how will they test? They'll often provide graphs with data. Okay, so when you encounter a graph, right, for enzymes specifically this topic, always ensure to firstly check your y and your x-axis very carefully. Okay, your y-axis, right, they tend to rotate or tend to mix up the title. So sometimes it can be, as you can see here, rate of reaction. Sometimes it can be rate of product form. Sometimes it can be rate of enzyme activity. So it really depends on the question. Just make sure you read carefully the graph and all its axes. Okay. Next, when answering such questions, right, using your own theory knowledge of these 4 factors is not enough. Okay, you must explain all these factors in context of the question to get your full mark. Right? So I'll show you some examples on how to tackle such questions later. Okay, so the main key point that I want to get across here is that explain each of these process very step by step. Okay, do not skip a single step. So I'm going to take an example of temperature. Okay, I'm going to briefly explain how do you answer. So let's say for temperature, the question asks, okay, how does temperature affect enzyme activity? Do not just tell me increasing temperature increases kinetic energy of enzyme and substrate molecules, resulting in increased enzyme activity. That will not give you the full mark. Okay, so how do you get the full mark? You need to first tell me, so listen closely. First, you must tell me the increase in temperature suggests an increase in thermal energy, which increases kinetic energy of enzyme and substrate molecules. Now, this increases the frequency of effective collisions between your enzyme and substrate, and hence increases the formation of enzyme-substrate complexes per unit time. This results in more products being formed per unit time and overall an increased rate of reaction. All right, so I went very slowly. If you missed out what I said, don't worry. This recording, this will be uploaded on YouTube, so you can always refer back to this video to revise. Okay, but my main point of what I just tell you the whole long story is that do not shortcut when it comes to these enzyme questions. Okay, go very step by step. All right, do not shortcut, don't skip step. Follow the explanation as per your lecture notes that your school give you. Okay. Then one more final point that I want to point out here before I move on would be that if you notice for the pH graph, as compared to your temperature graph, the range at which the rate of reaction spikes, right? Can you see? Okay, sorry, not range of reaction spike. The range at which there is enzyme activity is very narrow. So you can see it's over like that only in enzyme, whereas in pH, whereas for temperature, it's much longer. Okay, so take note, this is because enzymes work in a narrow pH range. So why is that? It's because if you remember from your biomolecules proteins, amino acids are amphoteric. So amphoteric, what does that mean? They contain both acidic and basic groups within the amino acid because of the carboxyl group and the amino group. So this allows amino acids to resist slight changes in pH, and hence enzymes as well, being globular proteins. Okay, generally most enzymes can resist pH changes within a very narrow range. So this is at most 1 or 2 of the optimum. 2 is a bit pushing it already. Okay, but most enzymes work within narrow pH range. They only can resist a narrow pH change, a slight pH change. Okay. All right. Oops. Okay, moving on to the next slide. Lastly, for enzymes, you need to know about inhibition. Okay, so know how each of the 2 inhibitors work. We talk about competitive inhibition and non-competitive inhibition. So know how they work and how they affect your Km, or otherwise known as enzyme affinity, as well as your rate of reaction where enzyme is fully saturated with substrate, otherwise known as your Vmax. Okay, so be very familiar. Competitive inhibition, we talk about competitive inhibitors, right? They bind your enzyme active site, competing with the substrate. Okay, so what does this suggest to you? Immediately, what should come to your mind is that competitive inhibitors, because they want to compete with the substrate for the active site, right? They are often structurally similar to the substrate. So as you can see on this diagram here, right? If you notice this blue little teardrop here, it has this like curve shape, which is very structurally similar to the substrate. Okay, so I can draw it out. So you can see, okay, this teardrop shape is also actually present in your substrate. All right, okay, I know my drawing a bit bad. Okay, but anyways, the main point here is that they are structurally similar. All right. If the question shows a substrate and another molecule that is structurally similar, so often they'll give you maybe the chemical formula lah. Okay, so then maybe this is your substrate is like that, something like that. I know it's very weird. Forgive my drawing, but main point here, you can see this is the substrate. This is the inhibitor. Notice that they are structurally similar. The question will point that out. So what must you infer from that is that the mode of inhibition is probably competitive inhibition. So you need to point out in your answer about the structural similarity between the substrate and this molecule that they put in the question. Okay. Now next, you can see that for competitive inhibition, what happens to the Km? The Km increases, but Vmax can still be reached at higher substrate concentration. Okay, so why? Because when at low concentrations, okay, low substrate concentration, when there is a lot of competitive inhibitors, so inhibitor concentration outweighs your substrate concentration, what happens? It is more likely that we get formation of enzyme inhibitor complex. Agree? Okay. However, why can Vmax be reached eventually as you increase your substrate concentration? Because as your substrate concentration increases, okay, it is more likely to form your enzyme substrate complex as opposed to your enzyme inhibitor complex. All right. And then hence Vmax can be reached eventually again. Okay, so this should be a revision for your explanation of how competitive inhibition affects your Vmax. Okay, moving on, we have your non-competitive inhibitors. Okay, now these are different. Non-competitive inhibitors bind at a site that you must specify is away from the active site. So what happens upon this binding? Okay, this non-competitive inhibitor will induce conformational change in the active site of the enzyme. So what does this do? Upon inducing conformational change in the active site of the enzyme active site will no longer be complementary to the substrate and hence will no longer be able to bind the substrate. Okay, so this is the most common explanation. Now, there are specific cases that I want to point out that upon binding of the non-competitive inhibitor, okay, substrate may still be able to bind enzyme, but the reaction will not be catalyzed. So the enzyme will not catalyze the reaction. Okay, but the more common one will be, is the first one I mentioned, that they will induce three-dimensional conformational changes in the active site. Okay, so Vmax in this case for non-competitive inhibition will be decreased. Why? Because non-competitive inhibitors, they reduce the total number of functional enzymes that can catalyze reaction. All right, but if you notice, the Km doesn't change because the enzyme affinity remains unchanged. Why? Because this non-competitive inhibition does not compete for the enzyme active site, which is unlike your competitive inhibition. So notice why for competitive inhibitors, your Km will increase is because they are competing with the substrate for enzyme active site. All right. Okay, so I hope this is clear for you. All right. Now, lastly, I want to do a quick note for DNA and genomics. So I don't have time to go through I hope this wasn't the part most of you came for. I don't really have time to go through DNA and genomics because it's a very long chapter. But what I want to drop is some tips for you. Okay, so firstly, what you see here on the left, this diagram, is what we refer to as the central dogma. Okay, what this means is just basically it's showing how genetic information is being processed in your cells, or basically how genes are expressed. Okay, so you can see that firstly, there's my annotation. Okay, you can see here, we talk about replication. Agree? Okay, so this is just your, you can see, remember, if you from your cell division in the interface, the cells will replicate their DNA. Okay, so you have to be familiar with the process of your semi-conservative replication. Okay, next, we follow by your transcription. So from DNA, how is the information converted to RNA into a message that can be read by your ribosomes? So this is transcription, using the DNA as a template to synthesize a strand of mRNA, messenger RNA. Okay, and then this mRNA, how does it give you a protein? It's through the process of your translation, carried out in your cytoplasm, carried out by your ribosomes. So I know most students tend to struggle with this chapter because wow, these three different processes are so heavy on the steps. Okay, what I recommend, okay, it will be firstly, I know lah. I know this is very dumb to say, but you have to understand the processes very well. Mainly, how do I advise you to study this is to understand the enzyme involved and the key steps. So what I mean by this is that instead of just jumping straight to studying this chapter, what is the first step of replication, second step like that, firstly, do an overall bird's eye view of each process first. Go and ask yourself, what are the key enzymes that are involved? For example, your replication, right? What are some of the key enzymes involved? We talk about your DNA helicase, or we also talk about, okay, not an enzyme, but protein. We talk about your single-stranded DNA binding proteins. Then we also have your topoisomerase, okay, with your DNA polymerase, with your primase, okay? So don't care about studying the steps first. Don't rush to memorize that. Learn each of the enzymes, what are the main functions. Once you're very clear with each of the enzyme and their functions, then you go into understanding the key steps. And this is the same for your transcription. So transcription, what is involved? With your RNA polymerase, we also have your, where's the DNA binding region? Your promoter, right? We'll talk about your TATA box, as well as your Pripnel box. Agree? Then we also talk about your general transcription factors. So understand these enzymes, proteins, or DNA binding regions first. Translation, we got what? We got your ribosomes. Then we also have your amino SLT-RNA synthetase, as well as your peptidyltransferase. We also have your codons. Okay, so you can see all the key players, all the enzymes involved. All right, then you go into each of the steps. So next thing to highlight, what is used as a template and what is the product? So as you can see here, for DNA, what is for replication, what is used as a template? The parental strand of DNA, correct? Because what we learn this is your semi-conservative replication. Then what about your transcription? We learned that your DNA is being used as the template. Okay, and what is the product? Your mRNA strand. Then we go down to translation. What is the template? Your mRNA strand, your mature mRNA, not your immature. Okay, your mature mRNA is the template. What is the product? Your polypeptide chain. Okay, so you can see, go very step by step. This will really help you to break down the concepts, and then it should be much more easy to tackle this chapter. All right, and then next, this one, most students tend to get very confused. Okay, go slow. Learn the reading frame. So one common way to one tip I have for you is that most of the time, what is being synthesized is often synthesized in the 5' to 3' direction. Okay, so this is the strand that is being synthesized. Let's say we talk about mRNA. If the mRNA is being synthesized in the 5' to 3' direction, then you should know that we learn in DNA, when we talk about double helix, right? Okay, we learned that we talk about what? Anti-parallel, right? 5' to 3', and then 3' to 5'. Agree? So if the newly synthesized mRNA strand is being synthesized in 5' to 3' direction, what should the template be? It should be read in your 3' to 5' direction. Okay, so this is how you approach this topic, because I know back when I was a JC student, I also get very confused by this reading frame. And what I found out was that trying to memorize, just purely memorize this chapter, right, it's not a good way to study it. You will get confused. Exams, if they were to test you essay question on these or any of these 3 processes, you might choke up. And then when you choke up, right, the whole thing goes haywire. So what I recommend is really go step by step. You can, for example, if let's say you get this as an exam question, what you can do is to write on the side as a draft, right, your enzymes involved and what are the functions. Then once you put everything out, once you lay all your materials out, then you start your essay in a very sequential manner. Okay. Then lastly, okay, videos help. So what I've prepared, okay, I've prepared 4 videos, which I will send in the chat now. Wait, how do I? Okay, I'll send 4 videos in the chat. Okay, these videos, right, very easy. Okay, you can just refer to. Okay, let me send the chat. Yeah. Okay, so I've listed for you. There is your replication, transcription, translation. Transcription got 2, and yeah, translation got 2 also. Okay, so these videos, right, how do they help? Very easy to find. If you don't know any bio concept, right, okay, one of your best friend besides your school teachers and your tuition teachers, okay, would be the videos on YouTube. Because what they do is that they will actually help you to condense or explain and visualize all these very complex processes really well. So I think one very familiar one is your Amoeba Sisters. I think a lot of schools use it. I think the videos are really helpful. They help as a way to summarize your learning. All right. Okay, so before we move on to practice examples, it's around 8 p.m. now. I'm going to give everyone a short 2-minute break. All right, so you can take a short 2-minute break and then come back, toilet break, drink water, anything. Once you're back, we'll continue with practice examples. All right, so, okay, everyone, please have a short break. Okay, I hope that was, I know it's a very short break, okay, but we're a bit tight on time. So, okay, so anyways, for those who missed out, hopefully you can just view the recording on YouTube, but let's continue with the practice examples today. So, for this section, don't stress so much about taking down the answer or anything. I'm going to walk you through the process of how you should be tackling these questions. So I think this is focus more on that. Okay, learn the skills rather than just the answer. All right. So let's start. I will go through each and every one of the question. Oh no, my camera is bugging out again. Okay, I'm just going to keep my camera switched off. All right, so first of all, cell bio. Let's go. I'll be doing practice examples for each of the each of the 3 topics. Okay, let's start with cell bio. As I mentioned earlier, very common that they'll show you an electron micrograph. First thing to do, right, is to always read the question context. So unfortunately here, they don't tell us whether it's a plant or animal cell. So you have to use your own inference, okay? How do I know this is an animal cell? You can tell by the general structure. It's like a sunny side up egg, right? As opposed to plant cells, which have a more rigid structure. Okay? So let's zoom in on each of the structures. P here, Q over here, R, this pair, and S. So notice that, first of all, right, for me, right, what I want to focus on is that, okay, off the top of my head, right, what immediately comes to mind? Okay, this densely stained region, what this reminds me of is you should be able to identify is your nucleus. All right, let's say this Q. Let's say I never study well, I don't really know lah. So I skip this one first. Okay, go to R. Notice that R points in a pair. Now, what do we learn in cell bio is that one of the special organelles exists in a pair, and somehow if you notice that they're actually perpendicular to each other. They look like a cylinder, right? This one is like laid down flat. This one is laid down perpendicular. All right. So something like, oh no, my drawing. Okay, something like this. Okay, so this is a pair of organelles. Now, what do we learn is this? This will most likely be, R will most likely be your centriole. Okay, a pair of your centrioles. Agree? And then lastly, with your S, S should be quite identifiable. Notice that there's a double membrane. One is here, and then there's all these folds. So S could be your mitochondria. Okay? So it cannot be your chloroplast. Why? Because, as I said, this looks like a sunny side up egg. It should be an animal cell, not a plant cell. So I already got 3, but I don't have Q, right? Doesn't matter. Let's look at the answers. P, nucleus, right? It should be, as I mentioned earlier, what's the function of the nucleus? Things to do with your genetic material. So P, DNA synthesis, yeah, possible. Polypeptide synthesis, not necessarily, because as I mentioned, remember, where are your polypeptides synthesized? In your cytoplasm by your ribosomes. mRNA synthesis, yeah, okay, possible. Contains genetic material. Okay, so only B is eliminated. Then we don't have Q, right? Let's move on to R. Okay, centrioles, right? What are they? DNA replication? I don't think so. Not necessarily. So C is out. Okay, that leaves your A and D. Let's look at D. Contains genetic material, yes. Q. Let's say we don't know what is Q. R, cell division. Yes, your centrioles are involved in cell division. S, Your mitochondria, ATP synthesis. Yes, okay. D looks very promising as the answer. Whereas here, they put S, absorption of light. No, that is your chloroplast. That is only in plant cells, and this is not a plant cell. So answer should be A. Sorry, not A, D. Yeah. Okay. So this is how you should be approaching electron micrograph. I know I did very step by step, but for those of you who are more seasoned, when you encounter this kind of question, this should be more of giveaway if you're very familiar with each of the different functions of your organelles. All right. Okay, moving on. Next question. Now, this is more of an application question, I would say. So for application questions, don't panic. I know there's a lot of things to read. Always go slow and make sure you fully understand the question context. Cells are basic unit of living organisms. Okay, yeah, obviously. What are the advantages of a multicellular organism being made up of many cells? Okay, let's go step by step. Which is true. Disabling one cell is unlikely to affect the functioning of the whole organism. Yep, this sounds true. Why? If let's say I have one cell that is dead, okay, will the whole organism die? No, because this is where we talk about growth, repair. Multicellular organisms have a lot of cells. If one cell shut down, the whole organism should still be able to function. So one should be correct. So we can eliminate D. Okay, cells of different types are genetically different. Huh? Decreasing the risk of expressing harmful recessive alleles. Off the top of the head, right here, this front part is already wrong. Cells of different types in a multicellular organism are not genetically different. Okay, they have the same genome, but what is different is the genes that are being expressed. Okay, so let's say your liver cell, this is the nucleus. The genetic material here, the DNA, okay, versus let's say your this is your liver cell, right? Liver. Let's say your lung cell. Okay, so this is your lung. I know this is a very bad drawing. Okay, so let's say your lung cell. Okay, the cell, the nucleus, the DNA, right? They are the same. Just that why do they look so different? It's because the genes that are being expressed in your liver cell versus your lung cell are different. Okay, they are not the same. Same genetic material, different expression. Okay, so 2 is false. Okay, we can eliminate 2. Okay, so anything with 2 is out. Okay, so that leaves B and C. All right, so yes, no. Okay, cells are able to specialize for specific functions, increasing efficiency. Yes. Okay, if you recall what I just explained, liver cells, lung cells, they all have different functions, but generally your liver cells are of the same similar structure for a specific function. Your liver, your organs, your lungs are for what? Gaseous exchange, right? Then your livers are for what? Your detoxification, stuff like that, metabolism. So 1 and 3 is correct. Next, just so this leaves B and C. Let's get 4. Damaged cells of one type can be replaced easily by differentiation of cells of any other type. Huh? Is this possible? If let's say my liver cell is damaged, can my lung cell suddenly differentiate into your liver cell? No, not true. Okay, I know this tests a bit on your stem cells, all right, but from the word itself, from the phrasing itself, it's a bit weird, right? Okay, this does not make sense because you're saying that any cell type can just differentiate on its own free will into another cell type just because it's damaged. No. All right, so what answer does that leave us with? Okay, that should leave us with C. All right, so you can see that how I approach this question, I took a very step-by-step process, assess each of the answers very carefully. Now, here comes the structured question that I was telling you guys about, not to get tricked. All right, so let's see. Read the question very carefully. These are my tips over here. Be familiar with all your cellular organelles and functions. Okay, figure 1.1 is the electron micrograph showing part of the liver cell. So this is an animal cell, synthesizes glycoproteins for secretion. All right. So what is shown here is not the entire cell, but part of a liver cell. All right. So this is already zoomed in. We can see that they're highlighting A and B. Okay, question. So this is from a prelim. With reference to figure 1.1, identify structure A. Structure A is here. Okay. So I hope what answer should come to your mind. Notice that A is a dense region within this bigger part. This whole thing is already a cell. Okay. So A, densely stained region. I hope what pops to your mind is your nucleolus. Okay, and additionally, what should come to your mind? So I never show it here, but the second part of the question asks what is the function. The function is that of your nucleolus, it contains genes coding for rRNA. It's the site of your rRNA synthesis, the site of your assembly of ribosomal subunits, your large and small ribosomal subunits, which I've covered just now. Okay. So why do I say don't get tricked, right? It's because I've seen students tell me A is the nucleus. Why? Because they think this whole thing is an animal cell. So they thought this is the nucleus. Okay, so you can see how schools tend to be a bit tricky. Okay, then they don't know. They thought this whole entire part is extracellular. Okay, so be careful. Read the question context. Don't just jump to conclusion. All right. Okay, moving on. Okay, so let's move on to biomolecules, some practice examples. Two of the isomers of glucose, alpha glucose, beta glucose, can be condensed in living cells to form a variety of compounds with properties suited to different functions. So automatically, we know this is talking about your polysaccharide. So you can see here, polymer of glucose, polysaccharide. Okay, which role correctly describes a polymer of glucose found in some living organisms? Off the top of your head. The moment you see beta glucose, you should think of cellulose. Whereas your alpha glucose, you should think of your starch and glycogen. So you should know function. We can straight away zoom into function. Okay. Beta glucose must be paired with structural support. So automatically, B is out. Agree? Then alpha glucose must be paired with energy reserve. So A is out. So you can see automatically I already cancel out two answers. So that leaves C and D. Okay, let's go one by one. C, branch change, alpha glucose. Alpha 1,4 glycosidic bond and alpha 1,6 glycosidic bond, energy reserve. Yep, this sounds good. What does this remind you of? This reminds me of your glycogen or your amylopectin. Okay, but let's check D just in case. Straight chains, okay. So we know beta glucose should be your cellulose, correct? Correct. But 1,6, we've only learned what? Alpha 1,4, alpha 1,6, and beta 1,4. So this is from nowhere. Okay, so D is out. Answer should be C. All right. So this is how you approach these kind of questions. So most of these questions so far are very recall questions, pretty straightforward. All right. Okay, next. Now, this is the more exciting part, your structured questions. Okay, so some of you may be looking forward to this. Okay, so firstly, same tips here. Okay, first thing, my first most important advice is always read your question carefully. All right. Be familiar with all the different biomolecules in terms of their structures, bonding, and function. So when I say biomolecules, I mean not just your proteins, but also your polysaccharides. So let's look at the question together. Following figure shows a three-dimensional model of the enzyme beta amylase. So we know this is a protein already. A small globular protein of 535 amino acids. Explain what determines the three-dimensional shape of such a protein. Okay. So first of all, one interesting thing I noticed is that they mentioned to me the specific number and sequence of amino acids. Okay, not sequence, but specific number. So automatically, this what comes to my head. Primary structure. Agree? Another keyword, three-dimensional model. This comes to my head, tertiary structure. Agree? Okay, so now why is it not quaternary? Okay, if you notice that, they're saying that it's one globular protein of 535 amino acids. They never say it's multiple chains. So off the top, and secondly, why is it not secondary? Can you observe any alpha helices or beta pleated sheets? No, I cannot see. Okay, so the answer should focus mainly on your tertiary structure and your primary structure. Now, I know it's 2 marks, but how do you go about answering this? Okay, I'm going to show the answer now. Firstly, the protein has a primary structure, which is the specific number 535 and sequence of amino acids that make up the polypeptide chain, held together by peptide bonds between amino acids. So notice, as I covered earlier, when answering protein structure questions, what must you have? Firstly, what is the structure level? Primary, secondary, tertiary, quaternary. Next thing you need to have, definition. So this is the definition. Specific number and sequence of amino acids. Lastly, what you need to have, the bonding. So here is peptide bonds between amino acids. Okay, so this is 1 mark. Because what is the second mark? Your tertiary structure. So the answer should be the polypeptide chain is folded into a tertiary structure, which is the same thing here. You can see it's the same format. Structure level, definition. Okay, and then the bonds. Between what? Between R groups of amino acids. Okay, so these two answers together will give you your full 2 marks. All right. So as I mentioned, please don't stress about copying down. Okay, anytime you can just refer back to the video. All right, so the polypeptide chain, the answer, make sure you are able to be very clear with your answer and address all portions of it. So this whole thing, 2 marks. Yes, I know there's a lot to write, but unfortunately, such is bio. Okay. All right, so let's move on. Okay, enzymes now. I'm going to show you an example here of how to approach this kind of figure question, as I mentioned earlier, right? First thing, same story again, read questions carefully. Be familiar with how and what factors affect enzyme activity. So I've already mentioned this specifically just now to focus on this. Okay, and take note of the phrasing as well. So how do you approach this question? Figure 2.1 shows the effect of increasing temperature on enzyme activity. All right, so you can see, first thing to take note, okay, I want to highlight. Students tend to see this, then they'll straight away say it starts at 0 or it's 0 degrees Celsius. No, if you can see here, there's actually some, it starts off with some activity already, around, let's say, 10%. Okay, and it's not 0, it's around, let's say, 3, 4, okay, degrees Celsius. So how do you approach this question? First of all, notice that what I did is to trace. In exam, please use ruler, trace to identify the values on the y-axis and the x-axis. All right, so as I said, just remember, be familiar, read the question carefully, check the axis. So y-axis is percentage maximum activity. Okay, so the values you can see around here, let's say, I think this should be 45 degrees Celsius. This should be 100. Okay, so this should be around 10% activity. This is maybe, let's say, 3 or 4. So exams, they will usually allow a range. So don't worry about not being able to hit the exact one, unless they give you like the grid lines. Okay. So this is what I have. This is what I interpreted. Explain why increasing the temperature increases the rate of reaction until it reaches optimum. So we want to describe, the question is asking us to describe from here all the way to optimum. Optimum is here, okay, which is 45 degrees Celsius. All right. So now follow closely, see how I answer this question. Firstly, as I mentioned, enzyme graph question, always make sure you're quoting the graph with reference to the diagram. As the temperature increases from 2 degrees Celsius, okay, 2 or 3 or 4, to optimum of 45 degrees Celsius, the percentage of maximum activity of enzyme or rate of reaction of enzyme increases from 10 to 100%. See, quote the data, quote the graph. Don't just jump straight to explaining the theory. All right. Then comes in your step-by-step explanation of the theory. I know this is 3 marks. You see how long is the answer? This is because as temperature rises or increases, the kinetic energy of enzyme and substrate molecules increases, and frequency of effective collisions between enzyme and substrate molecules increases. I'm going very slow. This facilitates formation of more enzyme substrate complexes per unit time, resulting in more product form per unit time. At optimum temperature of 45 degrees Celsius here, maximum number of enzyme substrate complexes are being formed per unit time. How can I tell? Because it's at 100% maximum activity. It can't go any higher. All right. So writing this entire part, I know it's very long, but this is what you need for your full 3 marks. Okay, everyone, I hope you are following closely. I know it's very long, very tedious, but the thing about bio is you need to firstly reference the figure, okay? And I want to make a point here. When I'm explaining this in context of enzymes, because enzyme questions tend to give you all these graphs and figures, but the point on referencing your figure stands for any question type, okay? Meaning to say, any topic, so long as they give you a figure and ask you to explain why or describe with reference to the figure, you must quote the data. So explain reference to lah. You can tell from its name. All right. Okay, so I hope you're following. Next part. Now, this is another interesting application question. It has come up before in some class tests, so I want to make a point to go through this. So enzyme X has a higher optimum temperature than enzyme Y. Suggest a reason for this. So how do you approach this question? First of all, let's break it down. They talk about enzymes. Agree? So enzyme, optimum temperature. So what should come into your head and recall is that firstly, when we talk about enzymes, we learn about what? Enzymes are tend to be globular proteins, or in the specific case of ribozymes, your RNA molecules. But when we focus on proteins, we talk about what? Tertiary and quaternary structure, agree? Because enzymes already have the unique three-dimensional conformation, like that. I know it's a very weird enzyme, with an active site. So such that it will bind here. So this is your 3D conformation, unique. Okay, so it can be tertiary or quaternary lah. Okay, next point. Question context. Enzyme X has a higher optimum temperature than enzyme Y. What does this mean? Okay, from your own interpretation, it should suggest that enzyme X is more thermostable at higher temperatures than enzyme Y, meaning to say that it takes a higher temperature before it starts denaturing. Okay, so I hope this is what you drew from the question context, because you can see if it's a higher optimum temperature means that, all right, if we were to put both at a very high temperature, it is most likely that enzyme X at, let's say, temperature of 60 degrees Celsius. Okay, this is where it reaches its maximum enzyme activity. But for enzyme Y, if we put it at 16 degrees Celsius, it is most likely past the optimum already, which is when your enzyme starts to denature and enzyme activity drops. Okay, so if we were to put it on the same graph, okay, it is most likely that enzyme Y has an earlier peak. So this is temperature. This is your activity, enzyme activity. Okay, so this is probably the optimum for Y. This is the optimum for X. 45. Let's say this is 60 degrees. All right, so you can see as it reaches optimum for X, the activity for Y decreases. Okay, so you can see this is so far all I've got from the question context. I haven't think of my answer yet. So what is next? Okay, what to think about next? Apply your knowledge. What is denaturation? What holds an enzyme's unique three-dimensional conformation together? So this is what you should be thinking about. And if you recall, to answer this second part, what holds an enzyme's unique three-dimensional conformation together? Basically, what I'm asking is, what are the bonds present in your tertiary or quaternary structure? Okay, so automatically, what are the 4 bonds you should remember? Your hydrophobic interactions. Okay, I'm not going to write out the whole thing. Your hydrogen bonds, ionic bonds, and then lastly, your disulfide bonds. Agree? Okay, so these are the 4 bonds present in your tertiary structure, and they is what holding your three-dimensional conformation of the enzyme together. Okay, where do these bonds occur at? Between the R groups of the amino acid that make up the polypeptide chain. So this, I hope all this is very familiar to you. See, it's just applying your knowledge, it's recalling. Now, what is interesting is that, okay, let's go to the next part. Okay, oops, let me clear the annotation. Okay, so what is interesting is that firstly, amino acids make up the polypeptide chain, right? Now, what is this amino acid over here? Okay, I know I accidentally flashed the answer. Okay, this is actually your cysteine. All right, so one thing that is special and you need to make a point to note is that while you do not need to memorize all the 20 amino acids, an important point to remember is that cysteine is the only amino acid with a sulf hydro group. Okay, so let me type it out. Sulf hydro group. Okay, which is basically your SH group. Okay, SH group. So what is significant about this sulf hydro group that is present in cysteine is that if you remember the 4 bonds that we covered, your 4th bond, your disulfide bonds. This is exactly where your disulfide bonds, some schools call it disulfide bridges, okay? This is where your disulfide bonds are being formed between 2 cysteine residues, okay, between the sulf hydro groups, okay? So I hope by now you're already piecing the answer together by yourself, okay? So what does this suggest about enzyme X as compared to enzyme Y? It suggests that enzyme X has greater number or presence of cysteine residues and hence greater number of disulfide bonds present in its tertiary or quaternary structure. Okay? And why is that? What does disulfide bonds have to do with the higher optimum temperature? It's because disulfide bonds are your strong covalent bonds and hence require large amounts of energy to break. We suggest that enzyme X is therefore more thermostable, because if they have more cysteine residues, more disulfide bonds, can able to exist and carry out its function at a higher optimum temperature. All right. So I hope that is very clear to you by now. Yeah, please don't stress about application questions. I hope you're able to piece this together. Just take it very step by step, and eventually you'll be able to apply the knowledge. Okay, so what I've noticed is that when it comes to recent years, H2 bio papers, be it prelims or promos or A-levels, right? Okay, there are less direct recall questions being tested. Most questions nowadays are testing more on, even though they are recall questions, right? Okay, they won't make it so obvious. Like they won't tell you like, oh, okay, recall what is the features of glycogen. Okay. Most of the time now they'll provide a lot of context. Your question papers tend to be a bit longer, unfortunately. All right. So what is the main technique that you really need to apply in your exams would be to really read the question context carefully, make full use of the information they give to you. Okay, and this is a very common application question when it comes to your enzymes and proteins. Okay, so I hope this has helped you by now. Hopefully this question come out for your promos, and then yay, you get a free mark here. Okay, all right. So moving on to the third part of my today's revision session will be some study tips. Okay, so this part you can relax a bit, not so much on content already, but please stick around because there's still the Kahoot quiz later. Okay, so some study tips going forward when you hit your J2 and preparing for your promos. Okay, now I know some schools have already started your promos, but bio most often the good thing is that they always schedule our good and bad. Okay, the good thing is that they schedule our papers usually after physics and chemistry, so you all have more time to study. Okay, so first of all, 1 week leading up to your promo exam, right? Okay, you should by now be wrapping up your content revision already. So it shouldn't be by now you're still studying, like trying to memorize contents that you have not studied. All right, so the key to really mastering biology is firstly, consistency. All right, going into J1, right, I feel that most of you will realize by now that compared to your secondary school bio, this is much more hectic in the sense that there's so much more content. And by the time you're done with one chapter and your school starts on the next chapter, with a whole lot more of content, you tend to forget your first chapter. All right, so how to avoid that is that as you learn each new chapter, always go back to the previous chapter. I'm not asking you to sit down and spend 1 hour to study and mark the entire chapter. Don't do that, you will burn out. But rather, really just flip through, maybe just like casually, informally in your brain, test yourself. Go and, for example, if you start on biomolecules, go and flip through your cell biology, okay, to really recap. Okay, just flip through. No one's asking you to sit down to memorize. All right, and this will really save up a lot of time when you actually sit down to properly mark and study for your exams when you go through the content. Okay, now the second thing that I want to highlight is to recognize when you need help. All right, so I can let me just be frank with everyone here. Memorizing bio, like let's say you memorize the entire book, but you don't really understand a lot of the content, right? You just memorize word for word. It can only get you so far. Okay, at most you can get a C or B at your A-levels. Okay, just to be very frank with you. Okay, so but it's good. Why I see all of you here today, right? Okay, it's because many students, they feel like, oh, bio, I don't need help, I just need to memorize. No. Okay, if you need help, go and seek out your teachers in school, go and seek out your tutors, all right, or go and Google online for videos and explanations. Okay, go and really understand the topic, and that is what sets apart your confirmed A students from your shaky B and your C students. Okay, it's because you guys aren't afraid to ask for help, to clarify concepts. It's not just about understanding, or bio is about just memorizing the keywords and language. Yeah, that's half of the battle. The other battle is to really understand the content. All right. Okay, so moving on. What can you do between your 1 week to, let's say, 3 days of your promos or your any other exam is to practice structured questions. Okay, if you know your foundation is not as strong, okay, practice more structured questions, meaning to say, for example, interpreting graphs, like the enzyme question I went through. Most of the questions I went through just now are the main structured content recall questions. Okay, these are the ones that really test you on whether you're very familiar with your content. But if you're comfortable with your content, you feel that you're strong, you're in a good place, then move on to trying application questions to if you're more confident, because application questions are often the ones that set the A students from the B students apart. All right. Now, 3 days to 2 days to your paper. Don't get lazy. Okay. By now, content should be done deal. All right. You should have been able to practice a lot of structured questions. You should be able to practice a lot of application questions already if your foundation is strong. At this point in time, firstly, don't neglect your MCQ. Okay, a lot of people think that MCQ is very easy. Yes, you should be getting full marks for MCQ, but you need practice. Okay. Then what else to practice would be your exam skills at this point, your time practices. So I noticed for a lot of students, my tutees especially, what they mentioned to me is that they run out of time for your exam papers. Okay, either because they don't really understand when they're reading the question, they're reading it, spending like so much time, 10 minutes just to read the question context and understand it, or they feel that they're writing a lot of points and then they still don't get the mark because they're writing out of scope. So all these actually point to poor management of time. All right. So you really need to practice yourself by taking time practices. Go and revise some by doing a paper from start to finish. Don't do a paper, 5 questions, then go and coffee break, go Starbucks and anything, then continue again. No. Sit down, try to be disciplined, set aside some time to fully do a full paper. Okay, and then this is already train your exam skills. Now, I want to make a special point. 3 days to your exam, right? Don't burn out. Don't stress yourself too much. Okay, I remember when I was a JC student, like 3 days to exam, I'm like on my bed studying until 3 a.m., 4 a.m. No, don't do that. That's very unhealthy. Okay, you will be very tired. Studies have shown that this one is really proof is out there. You can go and find. Okay, having lack of sleep and all that, right? If you stay up to 4 a.m. to cram the information in your head, right? Okay, it will not help you in the memory in the long run. You may find yourself panicking and choking during the exam, and then you won't remember key content. Okay, that's why I say this adds on to my previous point. As I mentioned, consistency is key. Okay, and then one day before your final exam, early full dress rehearsal. Okay, so what do I mean by this? So for those of you who are in art CCA, I hope you know what this means. It just means that maybe you can do just maybe one last paper, okay, one last time practice, and then right before the end of the day, try to fully recall the entire syllabus that's being tested. Okay, so I know for now promos, basically what I mean is that you recall the content for that is being tested for promos. Whereas when you hit your A levels next year, you just recall the entire the entire H2 bio syllabus. So you just casually test yourself, ask your health in the head. Okay, for cell bio, okay, nucleus, what's the function, organelle, blah, blah, blah. Are you able to do that? Okay, are you able to recall and answer all the questions, all the information? Okay, so one way to help you with this is that most of your lecture notes in school, they provide learning outcomes. Okay, so one way to really test yourself and do this full dress rehearsal is to look through your learning outcomes. Go and check whether you're able to address all the learning outcomes. All right. And then my most important tip that is often neglected by students is rest. Please go and rest, sleep early. Okay, don't stay up late and stress yourself. Okay, because staying up, as I mentioned, staying up late to cram the content, it won't help you. Okay, you will tend to forget more stuff. All right. Okay, so before I move on to the group tuition part, I want to shift over to the revision kit. Okay, why is my screen not sharing? Okay, yeah. Okay, so over here, I hope everyone has downloaded this. Later, I'll flash the QR code again at the end if you don't have it yet. So this is the revision package that comes for those who are attending this session today. And if you're viewing this on YouTube, okay, so what I've included is mainly, firstly, the answering technique. So whatever I went through just now, how to answer each question and the steps you take, right, is all covered here in the revision kit. So firstly, as I mentioned, you can see here, read question carefully, completely. There are 2 kinds of questions. Your factual recall questions will most often be, for example, define this, state this, name this, okay? These are just your recall question, how to answer over here, okay? And then for your application question, how to approach these kind of questions, okay? In case, as well as providing you with tips in case you get stuck. So let's say if you get stuck, just skip and come back later. All right. I've also included just one example of a factual recall question and how is the answering technique and process, which is what I went through today. Okay, but this example in the revision kit is different from the one in the slides because I wanted to give you guys more variety. All right. And then one application question example here. How do I go about really approaching the question? Okay. And then study revision tips, which is what I've already went through with you. And lastly, I think the ones that you guys are most happy for, excited about will be the summary of key concepts. Okay, so you can see here, I've included a lot of comparison table. These are by no means meant to replace your lecture notes. These are often more of just your summary, okay, to really revise. So let's say when you're revising, just go and look through, test yourself using all these tables and summary. All right, so I've covered mainly for the first 3 chapters of cell bio, biomolecules, and enzymes. Okay, so you can see here what I went through just now about the temperature over here. All is here. Okay, and then one last thing that is included in your H2 bio kit, this is actually taken from the SEAB A level, your H2 syllabus. If you notice at the end, right, of the syllabus itself, I've taken it, I just copy pasted it here. Here, right, they actually, for each of the keywords that can come up for exam, so you can see question types, define, describe, deduce. Okay, some students ask me like, oh, for define question, do I need to explain? Okay, oh, for state question, do I need to explain? All your questions are answered here. So you can see here, state implies a concise answer with little or no supporting argument, a numerical answer that can be obtained by inspection. So what does it mean? You don't need to explain. Most often this kind of question is 1 mark, 2 mark. Okay, so for example, just now I said state the organelle label A. Just need to write nucleolus. Don't need to tell me what is the function or that. All right. So I hope this helped. You don't need to memorize this. This is really just for you, for your own learning, for your own guide to really help you to understand better like how to approach your exam questions. Okay. All right. So I'm gonna move on to the last part. Okay. So, all right. So just some advertisements. So please stick around because we still have the Kahoot after this. Okay. OVERMUGGED, as part of OVERMUGGED, we're actually offering a group tuition, and we're doing the 2023 intake. So as you can see on this slide, we have all the different subjects listed on the left. Okay, different time slots on weekday and weekend. Okay, as well as various locations. Okay, so for me personally, because I have to see my schedule, but most often I'm teaching on weekend. Okay, I'm planning to teach open weekend classes, and weekday I'll at most open 1 or 2. It really depends on my schedule. Yeah. Okay, so QR code, and mainly for me, I'm teaching at I think Toa Payoh and Hougang mostly. Yeah, these 2 locations. All right, the rates, everything is here. So this all this information is included in the revision kit in the front part. Okay, if you're interested, I've included a QR code here for you to sign up. Or if you have more questions or that, all the information is in the revision kit, you can refer to that. Okay, I've included my Telegram here as well. If you guys have any questions regarding today's session, or regarding this group tuition, anything, you can feel free to contact me, or you can feel free to contact OVERMUGGED, the head tutor, Daryl. Okay, his information, the contact information, all that is in the revision kit, the front part. Okay, now I want to take few minutes to quickly explain this deal that we're offering. So 2023 J2 intake, right now we are offering this these 2 deals. Okay, firstly, you have your early bird special. Now, if you see, right, first thing I want to highlight is for early bird, meaning to say, right, if you really enjoyed my lesson today, you feel very comfortable with me teaching you, and you feel that you need help for your bio, you can feel free to, and you really want to sign up now. Okay, this is the early bird package. Is available now only until 16th October. So you still got some time to think about it. Maybe after your promos, you can think about it as well. If you sign up for the early bird package, right, what we have is that firstly, you get 2 free curated notes. So for example, you get H2 bio. Okay, so what do I mean by curated notes, right? I hope you guys saw just now. Okay, let me clear my... Okay, so I hope you guys saw this, the curated notes I've been using today to teach, right? So basically, these are notes that I've compiled personally for H2 bio from different JCs. Okay, credits to them. Okay, mainly what I did is to compile them. I've included mainly like the key information. I've removed some of the like for your own information so as to provide you a more focused study experience. Okay, so these are the curated notes. There is for other subjects as well. All right, so if you sign up now, you get 2 subject free. So meaning to say each subject got 3 booklets so you get 6 booklets total for 2 subjects you get to choose. So obviously one will be for bio 1 set. Then the other set you get to choose for free. Okay. Now next thing for early bird people is that you guys get priority selection for your classes and time slots. So for this, right, if you guys are signing up for this promo, please really indicate all your available time slots and days, and preferred one, okay, as much as possible, because for me, it really helps me with my scheduling. I want to accommodate as many of you as possible. All right. And then lastly, you get to receive a promos exam kit immediately. Now, what is this, right? Okay. I show you a sneak peek. Okay, so we have your, just now I showed you this earlier on is your revision kit. So this looks similar, right? Oops, why is it like that? Okay, so this is the revision kit. Your exam kit, right, is an expanded version of that. So you can see for your revision kit, I only did up to enzymes, right? Okay, but if you can see for your exam kit, I've included DNA and genomics, all the cheat sheets. So summary, transcription as well, okay, translation, all the way until your cell division. Okay, so basically you get two more topics, cell division, DNA, and genomics. And what else I've included? I've also included MCQ practice questions at the back. Okay, so this is mainly for my students who sign up for it. Okay, so wait, sorry, let me go back to the slide. Yeah, okay, so this is for the students who sign up directly for the early bird promo. Okay, you get this promo exam kit immediately. All right, so for you to really study. Okay, and then yeah, limited. So this is really until 16 October only. Okay, so my students will get that. Now, let's say you are not too sure whether you want to have work with me here. Okay, you're still figuring things out whether you need tuition. Okay, don't worry. We have another promo, is your trial package. Okay, so we get because tuition will start in November, you get to sign up for a free trial class. So try to sign up with this in your friends class, so it'll be more fun. Okay, you get one free trial class. You still get your 2 free curated notes. Okay, and then but at this point, because it's already a bit later, you can only select existing classes and time slots. So you won't have priority. You have to choose between what is already available. Okay, and then the lastly, which is the normal intake. So if you unfortunately, if you can't really come to a decision now, it's okay. No worries, no pressure. In 2023, anytime you feel like signing up, you feel like joining my classes, first trial lesson is always free. And then you only get 1 free curator note. So basically bio lah. Okay, and then the slots are subjected to availability because it really depends on my schedule and how many students I can take. All right, so I want to emphasize this is the deals are like there, very limited scarcity, urgency. Okay, so please, if you're really interested, please go ahead to sign up. QR code is here. If not, feel free to sign up in 2023. Looking forward to meeting some of you. All right. Okay, so some of you might ask, okay, what do I do in my H2 bio tuition, right? Okay, so most of my style, okay, for my tutorials, what I'll do is firstly, I'll go through content. So I'll do a lecture style. Then I'll go through, meaning to say I'll do it like, I'll go through the content today for the topic. So depending on what topic we're covering in class, I'll go through the key areas to focus on, teach a bit some of the more misconceptions, clarify certain concepts, and I'll give very informal quizzes. So it's like verbally, like I'll ask you, how do you answer certain questions and all that. What else do I do? I'll practice questions with you. This allows you to really test yourself and practice your answering technique. So students of mine will get resources, questions, worksheets to practice, okay, and with solutions as well. Occasionally, depending on time, I'll conduct class tests. Okay, so this is really to focus more on your exam skills and really to ensure that you get a holistic revision because they want to test all the concepts at once instead of just focusing on one chapter at a time. And then lastly, for my students, the extra perk is that you get free consultation anytime. So most of the time, a lot of my classes, students like to ask me questions. Okay, I'm available on WhatsApp or Tele. Most of the time, they'll ask me, I'll answer them. Or sometimes they want to ask me in person after class. I'm available. Okay. So tuties of mine, resources that I provide are, for example, your creator notes, as I said, is free. Okay, you get a 3 booklet of covering the entire H2 bio syllabus. You get supplementary material. So sometimes maybe I feel like you guys need more extra help. I'll give you, for example, summaries, stuff like that. And then I'll give you practice worksheets as well with solutions. Okay, so we will practice the questions in class. Afterwards, once you're done, I'll give you solutions as well. All right. Now, I know I focus a lot on H2 bio. So here also, I want to show the timeline of what you get when you sign up with me. Okay, so I go in phases. The first phase, when you sign up the early months, so let's say maybe November to December, what I want to focus on is to really revise your J1 content. I want to build a very strong foundation for my students, okay? Because going to J2, you'll be learning a lot of new stuff. I want to make sure your J1 content is strong. J2 will cover the J2 syllabus, okay? Then I want to support your school's teaching, your school's learning, okay? I know schools sometimes, because they're teaching such a big cohort, some of you may feel that you're left behind because you're a bit slower in terms of understanding the content. That is where I come in to help you really patch the gap and really teach you and clarify certain concepts. All right. I'm not here to teach you from scratch, but more to support your learning and any weakness you might have. Now, phase 3, this is maybe, let's say, middle of next year. middle of J2, I will wrap up the entire syllabus. So probably usually, depending on the class, I might be able to cover the H2 syllabus faster than your school. So with that, I'll wrap up whatever needs to be covered in the content, and then I'll switch into a focus on drilling your examination skills. So we'll practice a lot of questions, application questions will be focused on, and then exam skills, time management, stuff like that. Now the final phase will really be more of your own effort. Okay. Any questions you have, consultation, you can always ask me if you're my student. Okay. Really challenging yourself, bringing to me, asking me application questions. I will be providing challenging questions as well in my classes, giving you more application question. Okay, if you're ready for it. And then lastly, this is more personal for me. What's next, right? Meaning to say that, let's say some of you are interested in pursuing bio at uni level, NTU, NUS, overseas, up to you. Okay, being a uni grad, okay, I'm always here for my students. So I'm here to really provide you any advice you might have after JC. So some of you, maybe you're not sure whether you want to take this course in uni or that. I'm here to help you. Okay, but don't think so far yet. This one is more for after A's or nearing your A's. Okay. Now, I mentioned earlier I'm a H1 GP tutor as well. So I know this session was on H2 bio, but if you are interested and you feel that your GP is weak as well, okay, there's also a sign up for H1 GP. Okay, so for H1 GP, what I cover is paper 1 and paper 2, everything. Okay, I know some tuition centers or tutors, they tend to focus more on skills only. For me, I try to make time for everything. Paper 1, I know many students tend to say they lack content. So I have content lessons as well on top of essay skills. And then I also teach your compre, summary, and AQ. Okay, so my lessons for GP are more often discussion-based. Okay, so I would say that most students have fun with my GP lessons because it's very interactive. Don't worry if you're very shy also, you will learn to slowly step out of your barrier. I provide very exclusive compiled guides, and some of them created on my own, resources that really teach you how to approach AQ, summary, compre, SAQ. It's like a one-stop shop for everything. Really teach you how to approach the question, how to answer those questions. All right, and with GP, I feel that because in schools, your tutors, your school teachers have a lot of students, they tend to provide more generalized feedback. For me, because I'm working with a smaller class of you guys, I provide more targeted feedback. So I'll tell you, for example, your essay, what mistakes you tend to make, and what areas you can improve on, and so forth. And then same thing as for bio, I provide consultation. All right, so attached here is just some example of one of my own resources, a compre/essay reading that I've made, as well as one of my past student testimonials that I worked with quite a few years ago. Yeah. All right. Okay, so I hope everyone had a very good session today. Now we'll be moving on to one of the exciting parts, the Kahoot quiz. Okay, so get ready. I'm gonna flash the link, and then you guys can join. So only for first place prize. Okay, so for the first place prize winner, later please drop a private message to me in the chat, and then I'll DM you. You get me your I need to get your details so I can deliver the creator notes to you. Okay. Before that, I have one question. Hello, can you share the revision kit again? Okay, I'll share it after the Kahoot session. Okay. All right. So let me just stop sharing and change to Kahoot so everyone can get ready. Okay. Okay. Okay, can everyone see my screen? Okay, I know it's sharing the... Okay, yeah, okay, so I see 21 people here, so can start joining. Got creator notes to be won as first prize, huh? Okay, great. Okay, so try not to type some funny nickname lah. I need to track the winner later. Okay, so the first place winner later private message me on Zoom. Okay. So if you've generally done your revision to before the session today, or you've paid close attention to the session today, the question should be, some are easy, some are a bit more difficult. Okay, then we can see how. Okay, so come on, where are the remaining few? Okay, come on. Okay, once I hit 21, I'll just start. If not, I'll wait a few minutes. Okay, I'm gonna wait a few more, 10 more seconds. Last chance to join. 10, 9, 8, 7, 6, 5, 4, 3, 2. Oh, wait, one gone. Okay, maybe I wait for the last. Okay, can. Okay, I'm going to start now. Okay, everyone get ready, okay, starting. So, all the best, everyone. So, name the type of cell and label the organelles. Okay. 1 minute. Okay, one more answer. Okay, good. Most of you chose D. Okay, I'll go through each of the questions, don't worry. Okay. So as you can see here on the screen, you should be able to tell it's a plant cell. Why? Because if you notice, it's a very rigid structure. Okay. I'm noticing most of you chose B. Okay, animal cell. I think you guys got tricked because it looks like a sunny side egg, right? But it's not. If you notice over here, there's the sharper walls. Okay, so it's more defined in terms of structure. Okay, but in terms of the organelles, all right, D will not be your lysosomes. Do not be confused. Okay, I know it's a bit small. I apologize for that, but D, right? Okay, will be obviously your mitochondria. Okay, because you can see B is your chloroplast. Okay, so lysosomes are often much smaller. I know it's very high. I apologize for that, but if you zoom in, right, if you can see that organelle D has double membrane. Okay, so it can't be your lysosomes. Okay, I apologize for the very difficult to see. Okay, let's see. Good job, Exalia. Okay, next question. Okay, I see. I think this one slightly more easy. Okay, okay. Okay, this is a very tricky question. Okay, so first of all, right, okay, those who put active transport, let me mention why. So active transport, the focus is not on molecules that are too large to diffuse. So first of all, they already say active transport, the focus is more on against a concentration gradient, movement of molecules against a concentration gradient. And another key identifying feature of active transport would be that it requires ATP, energy in the form of ATP. All right. Now, why is it not facilitated diffusion? It's because, firstly, from the question itself, too large to diffuse in through a cell surface membrane. Now, facilitated diffusion, if you recall, it's mostly carried out by your channel or carrier proteins, all right, embedded in your cell surface membrane. So those are often molecules that cannot diffuse through on their own and require the activity or function of certain proteins as carrier or channel proteins. Now, the keyword here for why is it endocytosis is because it's too large. It cannot diffuse. So it needs to be taken up into a vesicle via endocytosis for it to go into the cell. All right. So I hope that clarifies. Okay, next question. Okay. Oh, catching up. Okay, let's continue. Okay, what is the function of the carbohydrate chain attached to glycoproteins or glycolipids? Wow, someone answered very fast. Okay, good. Most of you got it. To give the molecule a specific shape for cell-cell interaction or cell adhesion. Okay, very good. So when we talk about glycoproteins or glycolipids, the glyco just refers to your carbohydrate chain. So this is the protein, this is the lipid, depending on what it's attached to. Most of the time, the function is for your cell-cell interaction, adhesion, recognition, and so forth. Okay, so it gives a specific shape. All right. Now, most of you, the second most answer is B, to ensure that the glycoprotein remains attached to phospholipids. Now, this answer is not correct because if you notice that the protein itself, right, glycoprotein, in this context of the question, it's already embedded within the phospholipid bilayer, the cell membrane. So regardless of whether you have the glycol chain or not, it doesn't really matter whether, it doesn't really play a role in helping it to attach to the phospholipid. Okay? So because it's embedded already. All right. And then nextly, just to cover, ion channel, most of the time they will show it goes through. Okay, there's a channel. There won't be the carbohydrate chain. And lastly, defense mechanism. This one is factually not correct. Okay. All right. So let's see. Oh, Fitri, good job. Okay. Which carbohydrate has glycosidic bonds between carbon atom 1 and 6? Oh, that one person always very fast. So be careful, don't get tricked. Okay, one more answer. Okay. Oh, wow. Equally distribution. Okay, let me quickly go through this. Okay. Amylose, remember, amylose only has alpha-1,4. Okay, it's amylopectin that has alpha-1,4 and alpha-1,6 glycosidic bonds between your glucose molecules, your alpha glucose molecules. Now, your cellulose is beta-1,4. Don't forget. Maltose is alpha-1,4 between two glucose molecules. So maltose is a disaccharide. Okay, glycogen has both alpha-1,4 and alpha-1,6 glycosidic bonds. All right. Okay, I hope that's better. I hope that explains to you. Okay. Wow, Fiona, hot streak. Okay, schlonked. Okay. Next question. Okay, so what is the bond circle? So this is a bit application question, a bit trickier. So structure shows a glycolipid. Okay, which is the lipid part, which is the glycol part, and what is glycol? Okay. Okay. Oh, this is a very tricky question. Okay, so as I mentioned, okay, good job for those three, but don't be disheartened if you didn't get the answer. This was exactly a hard question, so I set it here. All right, so if you can see, right, question says this is a glycolipid. Agree? So I know that this over here, as you can see, the one I'm hovering my cursor at, that is the lipid portion on the right. On the left is your carbohydrate, correct? If you can see from its structure. Okay, there's carbon 1, 2, 3, 4, 5, 6. So you should know this is either this is probably your glucose. Agree? So glycolipid. Now I know I taught lipid is your ester bonds. Agree? And then I said for if there's a phosphate group, is your phosphoester bond. So why is this a glycosidic bond? It's because it involves the bond between a lipid and a glucose molecule, a carbohydrate molecule. Okay, I know in your lecture notes, they don't explicitly say that. They only tell you glycosidic bonds are formed between your glucose residues. That's why it's very specific, right? But this is why I mentioned this is an application question. It's not an ester bond because here, if you notice, this is not a glycerol backbone. Agree? It's not a phosphoester bond because this is not a phosphate group. Okay, and it can't be a peptide bond because there's no amino acid present here. So the best answer would be glycosidic bond. Okay, so if you want to know the exact definition of glycosidic bond, it's not actually restricted to only your glucose residues. All right, so it's a bit tricky. Okay, so good job. The rest, don't worry, take it as a learning point. All right. Okay. Combo breaker. Oh no. Okay, let's continue. Okay, another slightly tricky question. Wow, that one person really answered very fast. I'm impressed. Kahoot God. Okay, good. Most of you say primary structure. Okay, let me explain why. If all bonds in the protein are broken except covalent bonds, what are the covalent bonds that we've learned? Your disulfide bond as well as your peptide bond when it comes to proteins. Agree? Now, those who pick tertiary and quaternary, you might be thinking, oh, because there's disulfide bond is covalent, it won't be broken, right? But think about it. What about your ionic, hydrogen, hydrophobic? Even if your disulfide bonds remain, okay, those 3 bonds are broken. So that means that your tertiary and quaternary structure are lost. For secondary structure, remember, it's held by hydrogen bonds. So hydrogen bonds are broken because it's not a covalent bond. So that leaves your primary structure, which is held together by peptide bonds, which are covalent bonds and cannot be broken. All right. So primary structure will be unaffected. Okay. Good job. It seems like we have a front runner. Okay. Few more questions. So be careful. Graph on the left is temperature. Graph on the right is pH. Okay, let's see. Okay. So let's go through. Okay, I see most of you chose the blue, which is at Q, kinetic energy. Okay. Tricky question. At Q, right, for temperature, kinetic energy is not the highest. Why? Because temperature can still increase. The thermal energy can still increase, okay? By right, the kinetic energy can still further increase. It's just that what happens? Because the hydrogen bonds are broken. Because remember, past optimum temperature, the temperature gets too hot, too much energy. So what happens is that most of the time, your hydrogen bonds cannot withstand the heat and then it will break. But it's not saying that your kinetic energy is the highest here. All right. Now, why is it S? Is because as ionic bonds in the enzyme are broken. Yes. Okay, because remember I said I know I mentioned earlier pH tend to be more narrow graph. This is supposed to be more narrow, but past the optimum, what are the bonds that are typically affected would be your ionic bonds. All right, because we talk about acidic and basic when it comes to pH. Okay. Okay, next question. Oh, E is catching up. Okay. So competitive inhibitor, which substance is most likely the normal substrate? So are lipids the usual substrate or polynucleotide or what? Is it a bit tougher? Come on, 10 seconds left. Okay, so for this question, okay, good. Most of you realize that it's polysaccharide. Now, as I mentioned, why is it not polypeptide? It's because if you notice the structure, they aren't really amino acids. All right, so recall what are the identifying features of an amino acid. Okay, good job. This was an application question. Okay, nice. Okay, last 2 questions. So what is the most likely explanation for the observation below? A fungal amylase is incubated with starch at 22 degrees Celsius at pH 8. Sample is removed. After 5 minutes, mixed with iodine. Mixture is light orange. Okay, all the answers are slowing down. I think it's getting harder. Okay, let's see. The starch has been completely hydrolyzed to maltose is the correct answer. Okay, so it's not an inhibitor because if this is test you more on your recalling of the food test. Okay, iodine. Okay, if there's the presence of starch, it will become bluish black, right? Bluish color. But the mixture is light orange, which is the original color iodine, which means that starch is not present. You should recall amylase. What is the function? It breaks down starch into its monosaccharide, right? Okay, so meaning to say if light orange means that starch is no longer present. So inhibitor of amylase cannot be the answer. Optimum temperature is less than 22 cannot be the answer as well because it's saying that the optimum temperature of fungal amylase is less than 22 degrees. No, cannot be because it's already breaking down into all its monosaccharides already. So the pH is too high to allow the reaction to proceed quickly is the same answer as same understanding as the red answer. So it cannot be as well. Okay, so correct answer is that starch has been completely hydrolyzed to maltose. Okay. All right. Okay, last question. So I know that I didn't cover this. So this is like a bonus question for those who do a revision. Okay, I hope this isn't too hard. Okay, oh dear. Okay, I guess DNA and genomics is a weaker topic for a lot of you guys. Okay, so most of you chose D. Okay, so let's see. Okay, why is D not the answer? Okay, unwinding, yes. Helicase, good. Assembly of leading strand, yes. Polymerase. Now, filling in of gaps between new DNA fragments, right? Okay. Be careful. I know this sounds like ligase, but what this actually means is the removal of after your primers are removed, there is gaps between your DNA fragments, right? So what enzyme is filling in those gaps with DNA? With deoxyribonucleotides. It's your polymerase. Okay, so this is more talking about your lagging strand, your Okazaki fragments. Okay. DNA ligase, what it does is to fuse the two new DNA because when they put it in, they need to form phosphodiester bonds. So that is done more by your ligase. So answer should be B. Okay, so don't get tricked. Read question carefully. All right, so with this, congrats to the winner. Okay, I think it's like Schlongt, right? Okay, good job. Good job, everyone. Okay. It's like 9 out of 10. Okay, good. Okay, Ken. So whoever this is Schlongt, you can drop me a DM in the Zoom chat, okay, and then give me your good. So you can DM me your details, private. So for example, your name. I need your name as well as your address that we can deliver it to. Okay, yeah. Okay, so for the rest of you, let me just go back to my slide. Okay, so thanks everyone for joining today. Okay, so maybe Klong, you can stay a bit more. I will message you what information I need. Okay, so if not, everyone, thanks for staying with me and spending your revision time with me today. I hope it has been a very fruitful session. Oh, I know someone asked for the revision kit. Let me just flash it again. Okay, so let me share my screen. Okay, so this is a revision kit. So I hope everyone had a very useful session. So take note on the group tuition information I shared earlier. And I hope this session has been very helpful for everyone. If you guys are very interested in OVERMUGGED and need more resources, not just for bio, GP, or that, other subject as well, please feel free to check out OVERMUGGED. Okay, if not, all the best for your promos. I wish you guys all the best, really get the grades that you deserve and you want. All right. And I hope to see some of you signing up for my group tuition, be it for H2 Bio or GP. Yeah. Okay. If not, thanks everyone for the session. Okay. Yeah, have a good night ahead. Study hard, but not too hard. And yeah, I'll see you when I see you. Okay. Thanks everyone. Goodbye.